So, the next topic of the proteomics is application of the omic technology in bioprospecting. So, omic based technologies have many applications in bioprospecting, including culturing microbial communities, drug discovery, environmental remediation. So, culturing microbial communities, omic based techniques can culture diverse microbial communities without changing their composition. This allows for the characterization of new microbes, which is important for the scientific and technological advancement. Drug discovery. In drug discovery, omic technologies can be used to screen bacteria and microalgae for their potential as source of drug leads. This is done by focusing on their genomes, transcriptomes, secondary metabolite, biosynthetic pathway genes, and metabolomes. Environmental remediation. Omic based approaches can be used for environmental remediation. So novel product for production. Omic based approach can be used for the production of the novel products. Food safety. Omic technologies are used to detect the harmful substances in food. Ethnobotanical and phytomedical research. Omics technologies can be used to analyze and characterize biological systems in greater detail. This can open new perspective for the ethnobotanical and phytomedical research. This is the way of culturing microbial communities. So first to take the microbial community. Microbial community means this can be collected from any environmental samples, marine samples, tissue samples, any other. So from this microbial community, we have to isolate the community sample. So from that community sample, for example, marine sample, for example, drain sample, for example, polluted water sample. These are the community sample. Community means where they are located, where they are grown. So from that community sample, we have to isolate the strain isolation. From in that community sample, there are the different strains are there. So based on the different methodological processes, so we can isolate the different strains. From that strain isolation, we can cultivate the strains. That is called strain cultivation and conditions optimization. After isolating the strains, for example, bacillus is one type of the strain, another one is actinobacteria, is a soil bacteria, marine bacteria. So like that, different strains are cultivated at their conditions and their optimization. After optimizing the strains isolated from the different community sample, so from that sample we have to isolate the metabolite extraction. So this metabolite extraction, the metabolites may be proteins or maybe antibiotics or maybe peptides or maybe carbohydrates or from maybe nucleoproteins, different types. So this metabolites extraction is employed for the bioassays. Bioassay means which metabolite is there in that metabolite extraction. After bioassaying the different metabolites, they are used for the compound purification. Compound purification is done by TLC, preparative HPLC, etc. So by using the compound purification, these compounds are employed for the structural elucidation. Which type of the structure is identified and isolated from the different microbial community sample. So this structural elucidation have to be performed by using the NMR spectroscopy, HRMS techniques. By using the techniques in biomics, we can isolate the different peptides. This is the cultural microbial communities. So from this cultural microbial communities, there are different methods for identifying the sequencing DNA. So single strain, if you take the single strains are obtained from the genomics analysis, genome structure, function spaces and strain identification. So this can be achieved by using the different proteomic tools. Those are called the bioinformatics, chemoinformatics, gene mining, virtual screening. In gene mining and virtual screening, so there is in bioinformatic analysis, we can perform the metagenomic sequence, diversity and the species ID, metabolic integration. By using the metabolic integration, which type of the metabolites are isolated from the microbial community. And also by using the bioinformatic techniques, we can study the proteomics, glycomics and lipidomics, metabolomics and metabolic profile, SAR and functional elucidation. By proteomics means by using the bioinformatic tools, we study the proteomics. It explains the types of the proteins. And glycomics means what are the composition of the carbohydrates. Glycomic tool is there. And lipidomics, what is the membrane lipid composition. Metabolomics, which type of the metabolites are produced. So all these are studied by using the bioinformatics. This is the one of the tool of the proteomics. And also these tools also employed for RNA sequencing, metabolic pathway identification, differential gene expression, and also used for identification of the marker gene sequences, that is 16S rRNA, 18S RNA, transcriptional sequences. So and also community DNA. Microbial community, bacterial, which type of the microbial community we have to be used for isolating the single strain. Those are the bacterial microbial community, microalgae microbial community, fungal community. By taking the microbial community, different microbial community, bacterial community is different from the microbial community, microalgae community is different from the fungi. So this is the main community. From this community, two ways are there. One is isolation of the single strain, another one is community DNA isolation. This is the importance of the biomic. Another one is in agriculture field. In agriculture field, the application of the omics technology is plant material with variability in metabolite accumulation, different tissues, specialized structures, and elicited treated material, that is a jasmonate wounding and trichomes, latiferous, and different tissues. So this is the plant species. So in agriculture technology, so like as I said before that the microbial community, here is the plant community. Here we have to select the one taxonomy of the plant, one family of the plant. From this family, so we have to take the plant material with variability in metabolite accumulation. For example, leaf and flower, root and tissue, epithelial tissue, and also different parts of the material. Another one is, so this is the one tissue. So by taking the individual tissues, first one is multi-omic tools. So by using the multi-omic tools, we can study the transcriptomics, proteomics, and metabolomics. That means from this tissue, so we can study the transcriptomics. That means which level of the transcriptome is developed. From this transcriptomics, we can study the proteomics. That means study of the protein composition. From this proteomics, we can study the metabolomics. That means which type of the metabolites is produced from the tissue sample. So by analyzing all these transcriptomics, proteomics, and metabolomics, so next data analysis. By using the data analysis, phylogenetic analysis, and co-expression studies. In phylogenetic analysis, by using the sequence database, which species of this plant belongs can be studied. So next, candidate gene. Which type of the plant is belong to the candidate, and this, so we can study some. And next one is in the another importance, gene with known function in the pathway of the interest, genome sequence. By using the same plant, we can study the genome sequence by using the omic tool. Okay, so in this gene clustering mining techniques and candidate gene, here is the only isolation of the gene. We can identify the candidate gene, which type of the gene including in this plant. Another mechanism, by taking the whole plant tissue by mixing the whole plant tissue by extraction, here is a protein interaction. So here is a different parts are present in the plant: root, stem, leaf, tissue, flowering, so seeds. So which type of the proteins are interacted in the whole plant, so that can be also studied by using the proteomic tools. That is called the protein interaction. This protein interaction is studied by using the substrate ELISA techniques and microarray techniques. This protein activity can be detected by the GCMS analysis, LCMS analysis, and HPLC analysis. These over images are explained the overview of the approaches that can be employed to discover the unknown enzymes in the biosynthetic pathway of the interest. In A, identification of the candidate gene in plant material with the differences in metabolite accumulation through co-expression analysis or phylogenetic analysis to examine whether the gene clade is specific to the native plant producer. So this is the mechanism of the A. In B mechanism, mining genomic data to look for genes that are physically localized in the vicinity of the previously characterized enzyme. In C, total protein purification and fraction from the plant, followed by the functional assay and proteomic identification of the active fraction. So in this video, we can study the application of the omic tool in different ways. How can we apply it in the microbial community and agriculture community for identification of the new genes and the new species and unknown plants are which, and these unknown plants are which type of the genes are present in the unknown plants. Thank you.